So now it's time to delve into adaptive immunity. And when we're talking about adaptive immunity, the first thing that comes to my mind are antibodies. Big, important component of adaptive immunity. And antibodies are formed by plasma cells, which are differentiated B cells. And then there's also T cells in the form of cytotoxic T cells, helper T cells, and regulatory T cells. And combined, they are adaptive immunity. And so adaptive immunity is different from nonspecific immunity in that adaptive immunity is actually responding to specific pathogens that you're infected with, or even cancer cells. So let's go ahead and delve into this slide set and start checking it out. So here what we have here is a kind of a conglomeration of a variety of the topics. And so here, as I just mentioned, we've got antibodies produced by plasma cells, which are differentiated B cells. Most of adaptive immunity actually occurs in the lymphatic system. And what we're looking at right here is a lymph node. This right here is a three-dimensional structure or representation of an antibody. Over here is a dendritic cell, and we've already talked about dendritic cells a little bit when we talked about glial cells and also just phagocytic cells. And here we have a mama providing antibodies for her newborn. And so she is giving a type of passive immunity to the newborn. So breast milk has a lot of wonderful things, including antibodies. And so one thing that's important to understand is the 5 attributes of adaptive immunity. And specificity, as I mentioned, specific pathogens. Inducibility, meaning that you mount a response and it is induced only when needed. Clonality, well, you'll see that basically clone armies of certain types of immune cells are created, and that we will see in the context of clonal selection of B cells and clonal selection of T cells. Unresponsive to self, well, that means that largely you're not mounting an immune response to yourself, but autoimmune diseases are examples where this one essentially is violated. But in general, in terms of adaptive immunity, it is not responsive to self. And we'll see a very interesting aspect of that in terms of the topic of clonal deletion. And then memory, right? So why is it that if you get a vaccine, your immune system has a type of memory to respond to that type later on? And we will go into that. And so this right here is a little mnemonic that some students like to use for M-U-S-I-C, music. So if you can remember music and you remember what these are, that will put you on a path to memorizing and ideally then learning the 5 attributes of adaptive immunity. And so as I mentioned before, we're talking about B cells and we're talking about T cells. B cells are called that because they mature in the bone marrow. T cells mature in the thymus, which is also why people that have an autoimmune disorder against the thymus start having weak immune responses to viruses, for example. Okay? And that's because T lymphocytes are primarily responsible for essentially killing your cells that are infected or cancer cells, right? Whereas B cells, they produce antibodies for extracellular pathogens. So to talk about adaptive immunity, we really need to talk about antigens. And this is discussed somewhat in the innate immunity section, but let's get right into antigens. So antigens, if we look down here, there are 3 kinds. There are exogenous antigens, endogenous, and autoantigens. And autoantigens is what you have displayed on the surface of your human cells, right? So you have a lot of proteins sticking off the surface of your cells, so those are autoantigens, meaning your cell. Endogenous come from if a cell gets infected, for example, this virus. Well, basically what this particular cell does is it breaks up the virus a little bit and then puts little pieces of the virus on the outside. And you'll see that this is in MHC1 when we talk about the major histocompatibility complex. Now, exogenous antigens, exo, right, are just that, they're on the outside. And so when we're looking at this, make sure you go ahead and read all this as well. We're not going to cover it here. The lymphatic system is primarily where a lot of this adaptive immunity actually happens. Okay, and so I'm not going to ask you to memorize all of this by any means. you have an idea of what the lymphatic system is. Now, the lymphatic system, which is in the green right here, this is the lymphatic capillary. The fluid that comes into the lymph is actually coming from the kind of extracellular tissues, and that is fluid that leaks out of the bloodstream, gets absorbed into the lymph. And the lymph is not pumped by the heart. The lymph is essentially squeezed so that basically you have the ability to transport fluids and anything in those fluids throughout the lymphatic system. Okay, so B cells are primarily hang out in the spleen, the lymph nodes, and the tonsils, and in general, the T cells also hang out in the lymph cells. So what we have here is a lymph node, and inside these lymph nodes are, for example, antigen-presenting cells. And you'll see me refer to these as APCs. So go ahead and get that in your lexicon right now. So basically when the lymph is flowing, right, the fluid that originally came from your blood and is moving through extracellular tissues, it then goes into the lymph capillaries, the lymphatic vessels, drains into the lymph node so that any bacteria, viruses, any foreign material that was there now accumulate in these lymph nodes. So what we've got hanging out in here are antigen-presenting cells, as well as T cells and B cells. And so basically they are waiting for foreign pathogens to come on in so they can clear them out. And so when you've got bacteria, viruses in here, and you've got your immune system ramping up inside this, this physically gets bigger because more fluid comes in. You've got a lot of cells dividing. And so by checking somebody's, for example, neck or armpit for lymph nodes, and if they have increased in size, swollen lymph nodes means that you have an active infection that you are clearing out. So let's start getting into B cells and antibodies here, and all this ties together, but we need to talk about things in chunks before we can kind of bring them together. And so as I mentioned, they mature in bone marrow. That's why they're called B cells. And as I mentioned before, this is where they're mainly found, the spleen, lymph nodes, and the MALT, the mucosal-associated lymphatic tissue. And the mucosal-associated lymphatic tissue, there's a lot of it in your intestines. A lot of your immune system is in your intestines, okay? And a relatively small percentage of B cells are actually in the blood, although that's a very common thing to think of, and certainly there are B cells in the blood, okay? So the primary function of a B cell is to secrete antibodies. And so what kind of antibodies, what do antibodies do? So these 6 are now on this slide right here. And so what we're looking at is these red, goofy representations of antibodies. They're nice. Basically, if you ever need to draw an antibody, draw a Y, and that works quite well. And so antibodies work in a variety of ways. One way is that they neutralize the invader. So for example, if this is a toxin and it is physically bound by these, well, then this toxin is not capable of binding to whatever it binds to be a toxin. This virus, right, think about influenza, right? If you were to have hemagglutinin, if these were hemagglutinin, now they were bound by antibodies, well, that hemagglutinin would not be able to bind to the receptor on your cell and it would not be able to get in. Same with the bacterium. If an antibody coats a bacterium, then the bacterium has a hard time of getting into your cells. And so if we kind of look down here now, agglutination, this is kind of just a bigger version of what we saw up here where, for example, adhesin proteins were being bound to. So now what we've got with agglutination is you have so many different antibodies binding to these bacteria, and one antibody, for example, can bind to 2 antibodies, so they're actually cross-linking, and you can get a very large network of these bacteria. So it's so big that it is particularly easy for a phagocyte to find it and bind to it. Okay, so whether this neutralization is occurring or whether we've got the level of agglutination, which means that there's just a whole lot of these bound together, so it's a big mass. The key thing is that phagocytes will recognize the end here, the Fc receptor protein will bind to this portion of the antibody. You can call it a handle, right? This portion of the antibody right here is a handle that is recognized by a phagocyte, And so then it will take this in, bring this in, and undergo the 6 stages of phagocytosis that we talked about. And we talked about this in terms of when we, in the lecture of innate immunity, when we talked about opsonization. And as we mentioned there, opsonization is marking a pathogen with something, for example, antibodies for destruction. And what's going to destroy this particular pathogen right now? That's right, it's the phagocyte. Okay, so some antibodies actually have enzymatic activities where it actually forms hydrogen peroxide or ozone. You know, we talked about in the innate immunity, we talk about neutrophils producing bleach. Well, now we can talk about antibodies producing hydrogen peroxide. Pretty crazy stuff, right? And so basically hydrogen peroxide, you know, can kill bacteria, and so there you have it. And then this one over here, NK is for natural killer cell, natural killer lymphocyte, and we'll talk about this later. So the point is that if we go back to this slide, we talked about how some antibodies activate complement and inflammation in the innate immunity section. We just talked about neutralization, thereby preventing a bacteria from binding, right? If its adhesin proteins are bound, then you can no longer, they can no longer bind to a structure inside your body, right? And then opsonization, marking it for destruction by a phagocyte, agglutinization, really in a lot of the same ways, it's an opsonization form. And then this antibody-dependent cellular cytotoxicity, which is not formed on, which is this one right here. And then we've got the hydrogen peroxide formation. So if we just kind of wrap up this slide, then what we're talking about, B cells produce antibodies, and antibodies do this, which is demonstrated on this particular slide. Okay, so how do antibodies recognize antigens? Okay, so first of all, well, what is antigen? Antigen is typically a protein or a carbohydrate, and we talked about it a few slides ago when we talked about exogenous antigens that are on the outside of a bacteria, for example, in this polysaccharide component or protein or a particular toxin, as well as endogenous antigens that are virally infected. So they are pieces, they are parts of a particular pathogen. Okay, so basically what we're looking at here is a 3-dimensional representation of an antibody, and here is the Y that we can draw down here. Okay, so as I mentioned before, if you're going to draw an antibody, a good way to draw that is by drawing a Y. To get a little bit fancier, you would draw something like this, and that's a little bit closer to reality. And so what you can see is that this portion right here is that, right? And so this right here is the red, and the larger one here is the blue. And so ideally what you can see here is that an antibody is formed from a heavy chain and a light chain, and actually 2 heavy chains, 1, 2, and they're bound to light chains. So if you look at that and you're like, geez, well, I mean, that's a nice figure, but what does that actually mean? Well, each one of these are proteins. And so the whole structure is up here. And basically this light chain, which is the red, right? That's this right here. That's why it's smaller, the red. And then this purple right here represents this. Okay, so boom, boom. And so this right here is the constant formation. I called it the handle, and that is because the handle should be the same in all antibodies if a phagocyte is going to grab it. It knows that this is the handle. This portion up here, this is the part that's different. And that's why it's called V down here. It is the variable region. So there is the constant region, which is down here, and there is the variable region, which is up here. And this variable region, if we were to look inside there, this right here is now the variable region. So you can see that when we look inside here, we're looking inside here. So this is this, which is this. And what we're seeing is that this antibody is binding to an antigen. So this is another molecule which is sticking in there, and basically what we have is kind of a lock and key. We have this antigen binding site, which is part of the antibody, binding to the antigen specifically on the epitope. Okay, the epitope is the part that is physically bound. So an antigen has an epitope. An antigen has an epitope. This whole thing is the antigen, and there's more to it than that. We're just looking through this little keyhole, right, this little circle, and the part that actually binds is the epitope. Okay, so there are more than a million different kind of antibodies that are produced, and we'll talk about why. So basically, if an antibody binds to an antigen, this is the business end of it right here, this variable region. Okay, so basically, if we look at this particular epitope, this antigenic determinant, what we're looking at is a particular component. So just as we mentioned before. And so this is actually being a little bit redundant now, but this whole thing is the antigen, and this is a particular protein inside a cell membrane. And the epitopes, remember, are the parts that will bind to the antibodies. So this little epitope up here is being represented by that. Okay, so in other words, an antibody has the potential of being able to bind to this, or that. Okay, some pathogens have multiple epitopes on it. And again, some epitopes are on the outside, which is handy because if you can get antibodies to bind to a particular pathogen, then what this did is it just marked it for destruction, right? Same thing with this virus. If an antibody bound to it, and notice how I'm always drawing that variable region when it comes to what binds to it, right? If you look at where I drew this binding to it, it is not this part right here, right? This part right here is the handle. And the red portions, of course, are the antigen binding sites. So let's talk about how do we get, so now we know that there are multiple ways for antibodies, once they bind, for the pathogen to get cleared out of your system. That's what this slide is talking about. Now we can see from a three-dimensional perspective, okay, this is in two-dimensional, but hopefully you know what I mean, how antibodies actually bind to their antigens at the epitopes. So the question is, how do B cells, right, for a very specific pathogen, start producing antibodies? So let's talk about when a stem cell is then converted into a B cell. And what we're talking about is clonal deletion, okay? If we go back to here, we certainly want our antibodies to be able to bind to microbes, and we want them to bind to a virally infected cell so they can kill that. But what we do not want them to do is to start binding to proteins in your joints, in your brain, in your liver, in your thymus, wherever they are, right? We want B cells and their antibodies to not bind to self. So clonal deletion is the process where this is the quality control system. Okay, so now we're talking about the biogenesis of a B cell. And so what we've got here is a stem cell is then differentiates into a B cell. And what we're looking at, we're actually looking at different types of B cells here, and they're different types of B cells because what's going to be different is the component right here, right, the part that binds. That is going to be different on each one of these cells. So what we could do is we could, just to be clear about that, we could just go ahead and write in that this is one type, this is another type, this is another type, this is another type, and this is another type, okay? And what this is showing us then is that only, in this case, one type got through. And so what do I mean by got through? So B cells were just made. We have 1, 2, 3, 4, 5 different kinds of B cells. What happens is each one of these B cells is checked against autoantigens. So check this out. In your thymus, in your red bone marrow, what we've got is cells which produce every single type of protein that your body makes. Doesn't matter if it's a liver protein, doesn't matter if it's a brain cell protein. This cell makes it and displays it out here. It's basically putting it on the outside saying, hey, if you're a B cell receptor which binds to this, then we need to kill you, or you need to kill yourself, right? Because the only thing that should get through is a B cell that cannot bind through its receptor to something that is part of you, something that is self. So in this case, this particular B cell with its B cell receptor, and that's with this antibody that's sticking off the outside. And so let's go ahead and write that down. And so when we're looking at a B cell with what looks to be an antibody on the outside, this is called a B cell receptor. And you'll see that we will call this BCRs. And so this actually is just like the antibody that it would then make. Okay, so we know that right here, the variable region, this is where the epitope would bind, right? And so if this is a cell that then binds to all of these, or tries to bind and cannot, that means that it does not bind to any proteins that you normally make in your cell, and it is good to go, and it goes down to the blood vessel, and it careens off into the spleen where it can wait to see if there's any foreign invader which can bind to this. Remember, the key thing here is it's running a gauntlet, right? What B cells can get through the gauntlet and into the blood vessel and down to the stream? This one could do it because it checked out with its B cell receptor on every single possible autoantigen, every single protein that your body makes. I know it sounds crazy, but it's true, at least as well as we know it. Take a look at this one, which is labeled B. So basically, this had a B cell receptor which bound to an autoantigen. And let's just pretend that is a protein that is produced in your liver. So this is a liver protein that's being displayed out here. And this B cell receptor was able to bind to it, which means that if this B cell were to get through the gauntlet, go into the blood vessel, and go to the spleen, that means that at some point, if that B cell ever came into contact with your liver, it would start making antibodies against your liver. So you don't want that to happen. What do you want? You want this thing to die, 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 die. So it undergoes a process called apoptosis, which is when a cell essentially tells itself to die and kills itself. It's suicide. It's kind of like when we talk about neutrophils in innate immunity when it kills itself, although that suicide ended up forming a net, right, that neutrophil extracellular trap. In this case, it's basically just this B cell has the potential to bind to self proteins, so it needs to kill itself. So every single one of these B cells is going to go through the gauntlet, and the question is, does it bind to something that is self? If so, it kills itself. Does it not bind something to itself? It's just checked, thousands and thousands of different types of proteins. It didn't bind anything, so it made it, and it can go into the blood vessel, where it can then go into the spleen. So that encapsulates clonal deletion, right? They self-destruct, undergoing apoptosis, so they self-destruct if they react to autoantigens. Autoantigens are your proteins. That's what we have here. So you do not want your B cell receptors to be able to bind to the autoantigens, because then that means that you would have an autoimmune disease. So that is the quality control check for how you ensure that the B cells that are produced will not go after your own self. Okay? So then let's see how this particular works. So as I mentioned before, these are called B cell receptors. And here in this diagram, it's only showing one, right? And that is just to make it simple so that we don't see a whole bunch of things. The reality is that there's more. Okay, so this is showing, 7 B cell receptors. But check this out. There's actually over 500,000 B cell receptors on the outside of the cell. Okay? And that is because this is not drawn to scale, right? I mean, if it was going to be to scale, it would be tiny little pinpoints all covering the surface of us. But what you could think of when you're thinking of these particular cells, maybe you'd like to think of something that is a little bit more like here's the cell, and then there's essentially, it's just studded completely on the outside, and I can't draw any fine level because it's just so small. There are 500,000 of these just sticking off all over the place, right? And so these are just coated in these things, so that if they ever bump up against a particular, let's say pathogen, whoops, if it binds to, for example, a pathogen, right, then if it ever bumps up to it, if this interface right there is such that you have a bona fide interaction between the antibody and, for example, an epitope, right? So if this was now part of this larger bacterium and it bound to it, then it could get going on what we're about to talk about next. And so basically the point of this is why I started drawing this is that yes, there's 7 of this now to show us, but really there are 500,000 of that, okay? Every single one of these binding sites is identical to the next one. So this is exactly the same as this, which is the same as that, which is the same as that, that. So when we look down here, every 500,000 of these all bind to the exact same thing, okay? So it only recognizes one type of epitope. And so this is what we're seeing right here. So what I tried to draw in this interface right here is essentially coming out of, this is the B cell right here. And so the B cell receptor is coming out, and it's got the variable region, which binds to the epitope. And this would be, for example, this portion right here would be what I drew right there. So let's see how this works then. Okay, so we've kind of been building up to this. So in this particular case, we're looking at a B cell which has bound to a bacterial polysaccharide with a whole lot of repeating units. So every one of these are exactly the same thing. And so since these are all exactly the same thing, they can bind to a whole bunch of the variable regions of the B cell receptors if it's a good match. Remember, this is all predicated on the perfect match between epitope and B cell. And the fact that we've got millions of different B cells means that just by chance, there's going to be some type of B cell that will bind to an epitope of a pathogen, just by chance, because you make so many different kinds. It's a pretty crazy scenario. So what happens is after these receptors bind, it stimulates a signal inside the B cell and tells that B cell, you have just bound an invader. This is not natural. We need to clear this out. You need to differentiate into identical plasma cells, and then you need to start pumping out a whole bunch of antibodies so that it can bind to the pathogens and then clear it for destruction by the different methods that we already talked about. And so a B cell differentiates into a plasma cell, and this over here, this is pretty cool. This is a transmission electron micrograph, and this is showing the nucleus, and then it's got a whole bunch of rough endoplasmic reticulum, which are all those wavy lines, which is what we're looking at here. And so basically what we're seeing is that between the endoplasmic reticulum and the Golgi bodies, they produce proteins. And so this is going to just be essentially a factory of just pumping out antibodies and antibodies and antibodies, and that's what we're seeing here. So what happens? Antigen is recognized by antigen receptor, just what we talked about up there. The B cells clonally expand. The reason they're called clonally expand is these are identical to each other. They are cloned. They are a clone army. Plasma cells are produced, and antibodies are produced, which bind to antigens and then clears them out. So that is what we see here is there's no T cells here, so this is the T-independent humoral immunity clonal expression. That's a fancy way to say there's no T cells involved, and this is happening in the blood areas, the humoral. Okay, so that is T-independent. And so that is very limited in what it can do. It's pretty good, but to really talk about how your body ramps up an immune response with B cells, we need to get into some more in depth, okay? And so if we take a look at my hands up here, we can talk a little bit about the next topic, and that is major histocompatibility complexes. And so what they do is they are the mechanism, these complexes are the mechanisms which show epitopes to the outside of a cell. And we'll see that MHC1 is produced by all cells in your body. So right now, you have a whole lot of cells in your body that are digesting its own proteins, just little bits and pieces here, and sending them to the surface and then holding them out. It's as though this is a particular piece of a protein, and now I'm holding it out to the immune system so that something can test it. Okay, so that's MHC1. And that, for example, if I was infected by a virus, then one of the things that I break down inside myself would be a viral protein, so I'd be showing a virus protein. And that would be a cue for my immune system to recognize it and say, whoa, this is not correct, right? MHC2 are from, they're often referred to as professional antigen-presenting cells. And these are just the APCs that I've talked about, antigen-presenting cells. And so these are from phagocytes. If you ever see an MHC2 molecule, you know automatically that that came from a cell phagocytosing another cell, digesting it through the process we talk about in innate immunity, right? And thereby taking bits and pieces in the phagolysosome. We know how proteins are degraded into little chunks, right? So some of those chunks are then displayed on the surface of the cell. So what we're going to do now is we're going to take a little excursion into MHC land, major histocompatibility, and this was first discovered when skin grafts were being rejected from burn victims way back in the day, when this was still all new. Okay? And so the rejection has to do with these major histocompatibility complexes because they are presenting molecules and letting the immune system determine, are they foreign molecules or are they not? So let's dive into the major histocompatibility complex. So what we're seeing, first of all, is that major histocompatibility complexes are essentially these receptor-type proteins. You can see they're embedded in the cytoplasmic membrane. MHC1 has an antigen binding groove, and MHC2 has an antigen binding groove. Okay, and these will bind the epitopes. As I just mentioned, MHC1 is in every cell. I like to simplify it and just say every cell instead of every nucleated cell. And MHC2 is on B cells and all the other antigen-presenting cells. And so what that means is that the B cells also phagocytose invaders. So we'll talk about that in just a moment. Okay? So again, the reason we're talking about this is that for B cells to pump out lots and lots of antibodies and produce memory cells, which is a really important part of our immune system, we need to understand this particular system. So let's talk about MHC1 first. Okay? So again, all cells do this. So right now in your body, you have a bunch of cells that if we were able to peer inside, what we'd see is that normal, regular proteins, right, are being chopped up and they're being loaded into the MHC1 protein. And so once it's loaded up, it's now looking like this, right, because these are different pieces and parts of it. And then they're kind of zipped up into this vesicle. So now we've got this with MHC1 molecule with each one is a little bit different and binds to a different portion of the original protein. And so remember, each receptor, just like the B-cell receptor, each receptor binds to a different epitope. And so this vessel then is shuttled to the exterior of the cell where it binds to the cell membrane. And so basically what we have at this point is, if this is the, well, actually, why don't I go over here? If we look at up here, basically what we're doing is essentially we're putting these on the outside of the cell now. And so when we have this right here, I am now drawing it that is what we've got. Okay, so I guess I could be a little bit more specific and draw a little bit of the green epitopes. There we go. Right? So basically this happened so that this cell is showing off what it's got inside. Hey, hey, look at me. This is what I've got inside me. Am I just self, or am I something nasty inside, right? Oh, well, in this case, these polypeptides that went through this process, went through this vesicle, went to the surface and are displayed. Basically, no B-cell receptor, no T-cell receptor is going to bind to those because they're innate and say, you are in the wrong, we need to get rid of you, but that's what it's doing. It's basically saying, am I just me, or do I have something nasty inside of me that needs to clear out? If there was something nasty inside, then basically what would happen is we would have, essentially, this cell would have to be destroyed. Okay, so that is the purpose of showing these endogenous antigens to your immune system. Okay, that was MHC1. MHC1, all of your cells do this. to show off, am I normal or am I nasty? MHC2, what we've got here, is this again is for antigen-presenting cells, right, as well as B cells. And so we know about phagocytosis. We know that essentially, here's the phagolysosome. You've got all these little bits and pieces of proteins and carbohydrates. And basically what we've got inside here is we've got another vesicle that we've not talked about yet. And this is loaded up with MHC2, right? So what we're looking at is the same thing right here. So MHC2, and again, remember, all of these antigen binding sites that bind the epitope, all of these are different. And so they can only bind to something that is complementary. Now, what would be a good addition to this figure is to actually draw some of these, right? So now I'm drawing a couple of MHCs that essentially never made it, right? So they were here to begin with as well. Yeah, they're horrible MHCs, I know. Give me a break, right? But it's important to kind of note that you know, here, these MHC2s bound to epitopes, and they're going to be on the surface. Well, these didn't have anything that was complementary, so they just don't do anything, right? So not all MHCs will happen to be the right shape to bind to one of these. Only a perfect match will work. And that's a key thing for specificity, perfect match. So that's why I just drew these in there, because not every time do you have a perfect match. And so these are kind of the lonesome, well, they're not, yeah, little lonesome MHC complexes, right? So once this is displayed, okay, this is now saying, hey, an antigen-presenting cell, have just engulfed something, and now I am presenting it on the outside, and this is from a pathogen. So it is now alerting this. So now, to be consistent with what we looked at with the previous slide, this surface right here, right, where is that going to be? Right? If we look at that, what we're looking at here is essentially, we've got now MHCs, 2s on the outside, and they've got their epitopes, their antigens in the outside, right? So that's a commonality, whether we're looking at MHC1 or MHC2, the cell that had the MHCs degraded something and presented it on the outside. And it is basically, this is saying, I just destroyed something nasty inside of me, and these are all fair game, immune system. If you recognize these anywhere else, it's time for you to destroy them. And that is the purpose of MHC. MHC1 present endogenous antigens. So your normal proteins right now are being shown on the outside. Now your immune system won't recognize those as foreign, so no problem. However, if you've got something nasty inside of you, if you've been infected with the virus, then these are going to be showing on the outside, and these are what need to be recognized so that the foreign agent can be destroyed. Same thing right here, MHC2, they're exogenous antigens, and they're showing the immune system, hey, look what I've got, this is a pathogen, or cancer cells. And the reason it can recognize cancer cells is that sometimes cancer cells have mutated proteins in them, and so they are different, right? And so remember, anytime that an immune system recognizes an epitope inside an MHC as a foreign protein or a pathogen, that means that basically the receptor made it through this to begin with from the B cell. So let's talk about how this functions now with T cells. So, let's go back a few slides. And so what we're looking at here is we talked about T-independent humoral immunity. We said, hey, this is how antibodies can be formed. However, it's pretty weak, okay? To get more antibodies, right, we need to understand major histocompatibility proteins. So the question is now, okay, what does this have to do with all of what we just learned about major histocompatibility? Okay, so that is what we're going to talk about right now. And so for T cells, there are killer T cells, which are also called cytotoxic T cells, and there are helper T cells. We're not going to talk about regulatory T cells in this class. So now what we're doing is we're really looking at T-dependent humoral immune response. Okay, so remember, we already talked about how Plasma cells can be formed and produce antibodies, but we know that that wasn't a very robust system. This right here is the robust method for mounting an immune response. Okay, so this is T-dependent. And so this is the whole process on this one slide. I'm going to break it up into a couple of smaller slides. So basically this is now reproduced on this next slide and the following slide. So let's see what happens. First of all, this is T-dependent, so we know that somewhere in here there must be some T cells. Well, we're starting right off the bat with showing you how helper T cells are involved. And so what I've done is I've labeled these A, B, C, D, E, just to show you that there's different T cells, and each one of these T cells has its own T cell receptor. Okay, so things are getting a little complicated. We've talked about B-cell receptors. We talked about B-cell receptors. We've talked about MHC1 and 2, which also bind things. And now we're talking about T-cell receptors. Okay, so let's keep these straight in our mind. And so this is what is going to happen. These are individual helper T cells. This A, B, C, D, and E are not part of this process. What we're seeing is that there's an antigen-presenting cell. So let's go back. Antigen-presenting cell. Aha! So this process has already happened. So what's on this outside of this antigen-presenting cell? Well, remember, it degraded this, we'll say bacteria in this particular image. It then broke it up into bits. It loaded it into MHC2, fused it, and then put it on the outside. And it's presenting it. Remember, it's saying, hey, I just recognized and ate this foreign invader. Is there anybody out there that can match this up? So now this antigen-presenting cell is looking for a partner. It's looking for a helper T cell which can bind to one of these epitopes. So that's what's already happened. So this is the antigen-presenting cell. Yep, MHC2, just as we talked about, holding the epitope. So that's just what we were talking about. So if this epitope matches a helper T cell receptor, then it will bind. So think about that. We needed an antigen-presenting cell to have an MHC2 that could bind to the epitope. Now it needs, this antigen-presenting cell needs to just run into some helper T cell, which just happens to have a complementary T cell receptor that can bind that same exact epitope, right? I know this seems very far-fetched, but we're talking massive amounts of numbers of these different types of cells all in the lymph. And so the antigen-presenting cell migrated to the lymph tissue, and now it's checking itself out. Hey, this helper T cell, does this bind? Nope. Does this bind? Nope. This, nope. This, nope. This, aha, ding, 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 ding. We've got a match. We've got a winner. So now what happens, now that this antigen-presenting cell with its MHC2 loaded up with that piece of protein from that pathogen that it ate, now bumped into the right helper T cell that just happened to have that T cell receptor that is complementary to the epitope inside of it. Now what we have is a stabilized interaction, for example, with this CD4 and CD28. And there's more that's going on here. We won't get into that. But see how this physically binds to that? This means that we've got a stable connection and we are ready to go. So yes, this is an important connection. So what happens then is once this occurs, the antigen-presenting cell has a lot of things that go on inside of it. It is now activated. Okay, and so we can kind of look at it as why it's like throbbing right now. It is an activated antigen-presenting cell. What happens next? Well, basically, this causes this helper T cell to undergo internal components, right? So it is now being activated inside. So activation, activation, activation. And what this does is it then divides and divides and divides into multiple clones of the exact same thing. Very similar to what we saw with B cells. So this helper T cell bound to this epitope in this MHC2. The antigen-presenting cell underwent changes inside, and the T cell also did. The T cell got super activated, and its process is divide, 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 divide. Let's make a clone army of ourselves. And that's what we're looking at here. Okay, so So basically, and we don't need to go into interleukin-4, so don't worry about that, but there's a whole lot of chemicals that are being released by a whole lot of cells, and a whole lot more is going on, but we're just trying to keep it simple. And yes, this is relatively simple. Okay, so basically what happens now is this cell differentiates into a bona fide, full-on helper T cell, which then needs to bind to a B cell that also is presenting the same type of epitope. So you're saying, wait a second, Dr. Willett, this helper T cell bound to the same epitope that the antigen presenting cell, and we've got a B cell that binds to the same exact thing? I know it's hard to believe, but again, what we're talking about are sheer numbers. We're talking about a variety of, thousands, hundreds of thousands of different B cell receptors that each one of these will bind to a specific structure. And if it just so happens to be the right structure, then it's a match. Okay, so let's just assume that this is all true, right? Because there's a lot of evidence for it. So basically, this B cell, which has already engulfed a pathogen and is now presenting it, right? So if it engulfed a B cell, if it engulfed a pathogen and is presenting it in an MHC, what type of MHC is always the MHC from engulfing a pathogen? Is it MHC1 or is it MHC2? Right, so what would this particular be? Since it engulfed it from, it must be an MHC2, right? And if we look over here, we see that that is indeed the case, MHC2. Okay, so what we've got then is a B cell that binds to this helper T cell, and that's what we're looking at here. And if you look at it up here, it looks remarkably similar. That's because it is. So there's a lot of redundancy. So basically now, this helper T cell stimulates this particular B cell, which has the right epitope on it, which means that this B cell receptor, what do you suppose this is going to bind to? What do you suppose this is identical to? This is identical to the binding site here, which means that if we make a whole lot of this particular B cell, that's great because each one of these B cell receptors will bind to the particular pathogen. Okay, and that's what we're seeing here. So now we've got this helper T cell. It is now bound to this B cell and it is activating this B cell. Okay, so what happens when a cell is activated in this context? So what happened up here when this helper T cell was activated through a binding like this? What did it do next? Look at the slide. What did this cell do next? That's right. It underwent clonal division and clonal expansion and made identical aspects of it. What did the B cell back here do once it got activated? It made copies of itself in the form of B cells. So you can expect then that the next thing that is going to happen right here is that this cell is going to undergo clonal expansion, and that's what we're looking at right here, and it makes an army of plasma cells and it makes an army of memory B cells. And this is the payoff right here. This divides into a plasma cell, which divides into two. This divides into two. This divides into two, and it keeps on going, keeps on going. And if these start pumping out antibodies, up to 2,000 antibodies per second, each individual cell. That's crazy, I know. So, and what we're seeing here is this is an IgM and this is an IgG, and we'll talk about that in just a moment. So let's talk about what just happened then. What we've got is this B cell now, which has receptors which are complementary to the epitope, because that's the way it's built, right? Whatever is bound by this MHC2 right there is also the thing that can be bound at each one of these. And so then when this undergoes clonal expansion to make B cells, this no longer has the receptors on outside because they've done their job. This was the job of the B cell receptor, to be able to essentially, once it gets to this point right here, right, it needs to be able to bind, and then it will essentially form antibodies. And these memory B cells right here that are also produced, so basically this divides into a memory cell, and it divides into a plasma cell. Plasma cell will divide and divide and divide and divide, making millions of these, pumping out antibodies at a huge amount. And these will form memory B cells. And the memory B cells are the things that hang around your immune system months, sometimes even years later, which activates the immune system. So let's just go over this, a quick way, and now this is the whole thing all in once. I just went back a few slides. And let me just give you the basics of it so you understand the basic flow. What we had was an antigen-presenting cell. Before it got to this stage, it degraded a pathogen and then broke up its bits and presented it into MHC2, which is what we have here. It then is in the lymph tissue where it is going to interact with a whole bunch of cells, and by chance, if it binds into a helper T cell, which has a complementary T cell receptor, it binds. We have a good connection between other CD proteins. That stimulates the helper T cell to undergo clonal expansion. These are all identical. And so now we've got an army of these wandering around looking for the right B cell. So for example, if this bound to this one, well, that's not the right one. If this bound to this one, that's not the right. So these don't actually bind, they just bump into each other. If you do get a point where you've got a helper T cell receptor that is complementary to the epitope in the B cell receptor, then that binds and activates that B cell. That activated B cell then divides into a plasma cell and a memory cell. The memory cells go on to produce a lot more, and they hide out in your lymph tissue. And then the plasma cells over here, they're the business end. They're the ones that are pumping out antibodies for your immune system. And then the 6 different ways of how pathogens are destroyed are then occur. All right? So then if we look at the types of, so when we look at these antibodies here, right, how come there's 2 different kinds? Well, actually there's more different kinds. So now we know how antibodies first start getting ramped up into production. So it's pretty complicated. Think of the whole interaction between the antigen-presenting cell and the helper T cell, and then the helper T cell needing to go after it gets activated and looking for the B cell. You're kind of looking at it as a second opinion, right? If you've got these intense interactions between these 2 types of cells and then these 2 types of cells, and both of them are on, that's what turns it on. Instead of just having one switch say, go attack that, you need 2 switches. And that's beneficial primarily, well, one reason we think is because it helps cut down on possible autoimmune disorders, right? So imagine if one of your B cell receptors got through that could bind to, for example, a protein in one of your joints, right? Then if it just got activated willy-nilly and it started attacking your joints, that would be very deleterious, right? However, if you needed a 2-step process where before it could get activated, you need an antigen-presenting cell being stimulating and binding to something specifically with the helper T cell, and then that helper T cell then needed to go and activate that B cell. It just gives a much more quality control level on making sure you activate the right thing. Okay, so on the previous slide, we saw the IgM, which is the pentamer, which means that there's 5, and something that looked like just what we were drawing. And so different antibodies play different roles. And this is really important. Some, for example, one kind is only produced in breast milk. Others stimulate, for example, IgE will stimulate eosinophils to produce antiparasitic molecules. Right? Remember eosinophils, it's either allergies or it is some type of parasite infection which increases the number of those. So one way that they are stimulated to then produce antiparasitic molecules is for an IgE molecule to bind to it. Okay, so the other thing is that plasma cells, when they live for about 2 days, maybe a little bit longer, but the antibodies they produce can last for weeks. Okay, so and typically IgM is formed before IgG, etc. So in other words, there's a lot of complicated things that are happening, a lot of different types of immunoglobulins, which is what Ig stands for. And the purpose of this class is not to just go into depth on all of them, but to give you an appreciation for the complexity while still understanding some foundational aspects of immunology. So let's go ahead and look at these antibodies in this slide. And really the only thing I'm looking for you to know about this is that IgM is primarily the one that acts in complement activation, neutralization, and agglutination. So if you tie that together with those, right, that's important. So, and you'll see though that for example, neutralization and agglutination, that's a not uncommon one. Neutralization, complement activation. So there's multiple immunoglobulins that can do the same thing. The key one by far is percentages is IgG. So that's really the one that I want you to know, followed by IgM. The other stuff, it's interesting, I think, IgE, IgD, IgA, but for the purpose of this class, let's focus on IgG and IgM. Okay, so let's talk about, for example, a person that has been exposed to tetanus toxoid. That is tetanus toxin. And so, for example, this could be being injected with the toxoid as a vaccination, or this could be because you got tetanus. So what happens is there's a delay. And so there's a lot of reasons why there's a delay, but basically the first one that's made is IgM. And so what we're looking at on the y-axis is the concentration of a particular antibody in the serum. So first, through the process that we just talked about, so all of this just happened. Okay, so this happened once the tetanus toxoid was introduced into the person. It took a few days and then IgM was produced. IgM was produced first, and IgM does a really good job on agglutination as well as neutralization. And so you can see the toxin then is being bound by IgM this way. And then IgG is produced. Okay, so IgG is produced, and you can see that it goes in a higher concentration and it's produced for a longer duration. So now let's look at some day later on. So now basically what we're seeing in between these two particular pictures is essentially that time passes. Okay, so look at this as an example of a vaccination, right? And then you're exposed to tetanus toxin again, right? So you've had your vaccination. Look how long it took for IgM to be formed. It took 3 days, but it took probably, what, 7 days to get the highest maximum amount being produced. And then it took 15 days before you had IgG being produced. Okay, so that was your vaccination. Now, when IgM and IgG are being produced, they're being produced by plasma cells, right? And we see that right here, IgM, IgG. So what other types of cells are being produced other than plasma cells? So we know the antibodies are being produced, and that's what we're seeing here, IgM and IgG. So this just happened, and now we've got this. So what are the other types of cells that this produced? That's right, the memory cells. So when this was being produced, memory cells were also being produced, which means that the next time this person is exposed to tetanus toxin, right here would be, if we look at this and we look at this between day 6, right, half of that would be right here. That would be about what? Day 3? And I'm just looking at, I just split basically this in half. Right? So day 3, IgM is full on, whereas before, day 3 is when it first started. So we see immediately that IgM started almost immediately, and by day 3, it was rocking and rolling. Lots of IgM was being produced. So in other words, it started days ahead of the first time. So it is much faster. And look at IgG. Remember, IgG had this lag, right? This lag right here, from here to there, is no longer there. So IgG was hot on the heels of IgM, and within just a few days, your immune system is loaded up, and your body's loaded up with IgG and IgM. And look at what would have happened by day 7 on your first one. First of all, you've got some IgM produced, but hardly any IgG, and the concentration for this is much higher. So the secondary response, what we've got in just a few days is we've got a lot of IgM and we've got a lot of IgG. And that is really important, and that is the secondary response, and that is showing why vaccination is so important. And not only produces memory cells, well, let me back up. The reason it's so important is that you can produce a lot more antibodies a lot quicker afterwards, right? So more going this way and a lot quicker, meaning this has been shortened. And then all of this will occur. So in terms of immunity then, for example, active immunity, okay, is either naturally acquired, so this kid got chicken pox, right, and he got it from a friend in school. And so now this kid will build an immune response to the virus and essentially will be protected from it in the future. Or you can get a vaccination, and that's what we're looking at here. So active means that you have mounted this immune response yourself. You have produced these antibodies. You have undergone the process that we've just talked about here. That is active immunity. Passive immunity means you got those antibodies from somewhere else, right? Someone gave those to you, either your mom, right, or injected with antibodies. So sometimes if you're going overseas, you can get antibody injections, which will circulate in your blood for X number of days and will protect you. So passive means that you did not mount the response yourself. And naturally acquired, right? Both of these are natural because this kid just picked up chicken pox and this newborn is suckling from his mom. Whereas artificially acquired means that basically you got it from somewhere else completely. So we'll tie it a little bit of it together here with initial and booster shots. And what we're just looking at is antigens. We're not now breaking up antigen IgM or IgG. We're just looking at this one particular example of you get your first exposure. So this is your first shot for vaccination. You produce antibodies, and then later on you're given a booster shot. So what this is showing is you get a booster shot and the response is much more rapid, right? If you look at this from 7 days from getting the booster shot, you're way up there, right? You've got about 1,000 of arbitrary units of antigen, of antibody. Whereas before, after 7 days, you were just getting going. So we see an increase, so it's faster and it's more robust. And that's showing why booster shots are so important. And then in your bloodstream, look at that. You're holding on to those antibodies instead of there being none. And the reason we've got this red curve is to just show you that if at the same time you also get a vaccination for another disease, well, that particular disease needs to start over. In other words, a vaccination against one particular pathogen is not going to give you protection to another one. So this is showing that if you get a couple of vaccinations at the same time, one was a booster, that's going to boost your immunity in a lot of ways, both shortening in the time as well as increasing the amount of antibodies. But for the new shot, the new vaccination that's not a booster, you still have the same type of response that you had before. And so that is a primary response and a secondary response. And this is a primary response. So that is what we're going to talk about for immunity. And that is really a good place. You may have already taken a few breaks now, up until now, but this is a good place to break as well if you want to come back later and watch the remainder, because what we're going to get into now are cytotoxic T cells. So if you want to pause it and come back later, now's a great start. Otherwise, let's just get into this. So killer T cells. If we look over at this picture, this right here is a killer T cell. The C is for cytotoxic, meaning it kills cells. And we see that it's binding to a cancer cell. So killer T cells will bind to cancer cells, which is great. This is when your body rids itself of cancer by itself, you can thank your killer T cells. And also, if you've got, for example, virally infected cells, your killer T cells will do that. So basically, killer T cells destroy cells directly. That's what we're seeing over here. When we talked about antibodies, that was an indirect method. It was marking those pathogens. Here, this cell is just going to tear this cancer cell up and kill it. Okay, so, and this particular cytotoxic T cell is specific for this cancer cell. Remember the whole specificity thing. And so you know right off the bat that there must be something going on in the interaction between these. And if you're starting to think about major histocompatibility interactions, well, you're right on target. And so yes, here's another one of these dreaded diagrams, but follow with me and let's just get to some of the very specifics. So first of all, activation and clonal expansion, These are terms that you now know. We just talked about it. We know that if you're going to activate something, you need a specific interaction between major histocompatibility complexes and T-cell receptors. And that's what we're seeing here. This right here is a dendritic cell. That's what DC stands for. And so dendritic cells, they have the good fortune of being able to produce MHC2 as well as MHC1. Okay, so wait a second. Let's take a moment to remember which do which. So MHC1 are all cells make this, and they present just various types and bits and pieces of proteins that are inside. Whereas MHC2 are only produced by antigen presentation, and they're showing in epitope form what was digested. So I ate something nasty, and I'm looking for a helper T cell to interact with. Okay, so that's what we're looking at there. And so if we look at this, let's just keep it simple for the purpose of this class. You don't need to know about interleukins. You don't need to know about what these arrows are doing, but they're basically jacking each other up. You can look at it that way. Okay, so the dendritic cell, through a specific interaction, it ate a particular viral infected cell. Okay, that's what it did. It ate a virally infected cell. And some of the viral proteins are now being displayed on here. It activates the helper T cell just like we talked about before. Okay? But also, it is now presenting epitopes through the other MHC. And what it's doing is, if there's a match between this T cell receptor and the MHC1 of the dendritic cell, if there's a match for that epitope, right? If the T cell receptor can bind to that epitope, then what's going to happen to this? What happens once you have that interaction and you've got a nice tight bond? This is going to activate. So it's going to activate, and what happens when a T cell or a B cell gets activated? It clonally expands. So it's the same exact topic, so don't get too worked up over this. I'm not going to start getting into a whole bunch of detail about CD8 and MHC1 for all this. You do need to know the difference between MHC1 and MHC2, but if I'm going to ask you a nice essay question on describing this, you don't need to have all that sorted out completely to still get a really good grade. What you do need to know is that after this interaction happens, you activate this so that you then have activated killer T cells as well as memory T cells. So this is very analogous to the T-dependent humoral immunity. And this is a T-dependent cytotoxic T cell immunity. And so basically, these are not going to produce antibodies, right? They're not plasma cells. They're active cytotoxic T cells, and now they are dividing and dividing. And now what's going on is these will leave the lymph node, and they'll go look for the foreign invaders that they need to kill. So in other words, this dendritic cell chomped on a virally infected cell, or a cancer cell, but we'll just say a virally infected cell for this particular thing. It then got into the lymph and went back to the lymph node, where it then checked for killer T cells and helper T cells. And it said, hey, anybody here match these epitopes I've got? I just ate something nasty. This is a virally infected cell. And this helper T cell bound and said, yes, I'm a partner. Yes, I'm a partner. We both can help you out. The helper T cell then activates this inactive T cell. So by binding here, this killer T cell by binding to the MHC1 with the epitope in it, and then by getting activated by the helper T cell, this gets supercharged, it gets activated, and starts forming a whole lot of killer T cells, which will then go out and leave the lymph node and do some killing, as well as memory T cells, so that you can still have that memory response. Remember, for B cells, this is a great thing about immunity. You want that to ramp up. Well, now we're talking about killer T cells. There's going to be memory cells as well, so that if you've got this virus coming in and infecting other cells, 2 years down the road, these memory T cells will be activated. And the great thing about these memory T cells being activated later on is that you don't need all this stuff to happen to begin with. So memory T cells can just get jacked up by helper T cells, and you don't need this process to happen anymore. All right, so I believe we talked about this, but don't worry about interleukins. And this right here is showing the interaction between the lymphocyte, and in this case, cytotoxic T cell, or a helper T cell, I don't know from the diagram of this, and the dendritic cell. Okay, so basically this right here is being represented here, either that or here. So there is, right, truth to this. And so how cool is that? It's being activated. So this dendritic cell is activating the T cell based on what it chomped up and what it produced and is now presenting in its MHC1 and its MHC2. Okay, so now we know how we go from a virally infected cell that's been chomped up by a dendritic cell results in the production of killer T cells, which now leave the lymph node and start marauding around. So what happens to actually kill virally infected cells? Well, this is now the activated killer T cell that we had from down here. Okay? It's all jacked up and it's ready to roll. And so what does it need to find? Right? It needs to find an epitope that it can bind to. It's all about that specificity, right? I mean, it's tough to learn this material, but basically you know that this cytotoxic T cell, the reason it got activated is because it bound to this MHC1 that had a complementary epitope. So it internally then knows how to make, right, these T cell receptors to then bind to that virus. And so now you've got all of these marauding around, and there's not just 4, we're talking about probably hundreds of thousands. They're marauding around looking for the epitope that will bind right there. So if it does bind to a cell that's been virally infected, okay, so let me ask you this. This virally infected cell, this could be, let's say this is a kidney cell. This kidney cell was infected by a virus. What did it do to the virus just as a course of action? It broke it up into pieces, right, and then loaded it up to there. So if we go back to what we discussed for major histocompatibility, basically it chomped up a bunch of these epitopes from the virus and it displayed them, right there as the viral epitope. And so that's when we talked about MHC1. So now this virus, which has never interacted with the cytotoxic T cell, it's just, I mean, this virally infected cell, it's just in the kidney doing its thing and it's been infected and probably more viruses are leaving. But if it hasn't yet and it's broken them up into these little bits, well, it is presenting them in the MHC1 protein. And remember, it's only showing one for the sake of simplicity, but don't get so hung up on that because basically, remember that this is happening on a large scale. And so what I'm drawing now are other MHC1 proteins, right? And inside each one of those is a particular epitope from that virus. And so each one of these, you know, and some of them will be the same, right? But some of these will be very different from each other, and they came from inside there, right? So that's what we're looking at for all of these. So what we're seeing then is essentially this virally infected cell is now coated with MHC1 all over it, and if it then bumps into a cytotoxic T cell that has the complementary T cell receptor that can bind to the epitope inside the MHC1, so in other words, another active cytotoxic T cell could come over here and bind there and cause an interaction. Another one could bind here. Another one could bind there, just so long as it has been activated and it is the right shape. Okay, so now that this has happened, right, it's taken a long time to get to this point, so now our cytotoxic T cell, our so-called killer T cell, has bound to a virally infected cell, and it knows that this is a cell that must be destroyed because it is sensing, it is binding to this viral epitope that this cell was born to destroy, right? That's why it exists. If it ever interacts with a cell that's got one of these things sticking out of there, its process is to kill, 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 kill. So how does it do it? It does it in 2 different ways. One way is, and so now if we look at this, that's right here. This cytotoxic T cell is right here. And so we're basically blowing up this particular region. Right, so this is now what we're looking at there, right? And so we can see that this belongs to that, this is that, that's that. Okay, so how does it kill? Basically, and this is really, well, basically, this is pretty crazy. So inside the cytotoxic T cell, and maybe we could just kind of prelude that a little bit so it doesn't look like it's just kind of coming out of nowhere. What if we had, because it does, a whole bunch of these. So now I'm trying to show these right here. These are vesicles that have perforin in one and granzyme in another. Know these terms, perforin and granzyme. So what do you suppose perforin means? Think about perforate, right? So check this out. When this is activated, right, boop, 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 I'm recognizing a viral epitope on the surface of a human cell, must destroy, must destroy, must destroy. Basically, it sends things out to this virally infected cell to do the dirty deed and kill it. That is in the context, that is called perforin. So these vesicles release these perforin molecules and they form this pore. So once you've blown holes in there, and so that's what we've got here, we've essentially got holes that are being produced. So what we've got here is a hole, a hole, a hole, a hole. That's what the perforin is producing. And so this perforin right here, complex I'm showing is there. And so this granzyme then is released, and now you've got these enzymes which then go in. So then the next step though, if we look to here, is essentially, now this releases a bunch of these perforins which will get into the cell. So this releases a cloud of these granzymes and they go into. And so by being able to go inside the cell, they now have access. So basically what we've got is a very elegant way to punch holes in this virally infected cell and then send in along the, well, not a suicide squad, some type of army. These are enzymes. So granzymes activate apoptotic enzymes. We talked about apoptosis. Basically that means that the cell is now going to do what? If it undergoes apoptosis, what does it do? It kills itself. So basically, this cytotoxic T cell punched holes in this in the form of perforins, sent granzymes in there, and these granzymes then make this cell kill itself. And so basically what we then have is a nice way to essentially destroy this cell through perforins and granzymes. And then there's another way, but we don't need to discuss that for the purpose of this class. Just know the discussion on perforins and granzymes. So what stimulated this cytotoxic T cell to do that? It's all about the interaction between the T cell receptor and the viral epitope in the MHC1 protein. And that is how a cell-mediated response destroys compromised human cells. Yeah, so we'll leave it at that. So now we've talked about, here we go. This is really what to leave it off on. So as I mentioned, this infected cell can have a bunch of particular cytotoxic T cells binding to it, just so long as there are MHC1 proteins that have an epitope that they can respond. And check this out. This is a cancer cell down here. And each one of these, this is crazy, this is a killer T cell, right? So it's physically bound on. And so what's it doing right now? What is it? So now that it's bound, right, this interaction has occurred. So now it is releasing perforin and granzyme. And so, for example, we can see a hole there. Here's another example of a cell that is being destroyed. And essentially, look at the perforin, just blew it open. And sometimes so much perforin can go in as well that water will go in, just like we talked about before. Water can flood in there as well and cause osmotic lysis. But the immune system isn't leaving that to chance. It is sending in these granzymes so that it will then essentially cause this cell to die. And that's what we've got. So again, when you're looking at this material, right, think about it in the grandiose way that really, I don't know, it's pretty exciting, right? These are marauding around, they've latched on, and they're sending in this crazy payload to actually kill the bacteria, kill the virally infected cell. And so that is your how cytotoxic T cells interact. All right, so that was a lot of information in this lecture, right? B cells, T cells, MHC1, MHC2, memory cells, helper cells, killer cells, granzyme, perforin, primary immune response, secondary immune response. A good approach to this is to take these into chunks, learn the chunks, and then bring them all together. In addition to this long lecture, there are animation videos on Blackboard, which you should definitely spend the time with. You will see that there's an overlap, right? If I did not cover it in this lecture and it is in one of the smaller videos from the animation, I'm not going to hold you accountable for it. The stuff that's in this lecture is the most important. The other animations are to help reinforce and to learn this, because it gets pretty crazy, right? There's a whole lot of different topics, a lot of nuances. Take to the discussion board if you have any questions, right? That's what it's for. So I hope you enjoy this material. When students learn this in person, it is one of the more challenging sections just in general. And so students that even don't use flashcards, sometimes will use flashcards for this material to kind of get everything in their mind, sort it out, as well as to sort out what's innate immunity and what is adaptive immunity.